Let's chop into action and look at the physiological effects of light versus shade in agricultural crops. Is agroforestry an option for basil and tomato plants? This experiment examines the potential of growing agricultural crops at lower levels of light. If our plants are able to grow under shade cloth, then it is likely that farmers could plant tomatoes and basil underneath tree crops to improve overall land productivity. A perennial herb, Prunella vulgaris, was also included into this study to test if native species with different life histories may have an advantage surviving in the shade. We measured leaf chlorophyll content, internodal length, stomatal conductance, and stomatal density. Our results will discuss how shade treatments affected these qualities. Unfortunately, our shade cloth treatment was much darker than the understory of a forest, so many of our plants experienced fatalities. If we were to repeat this study with more resources, we would have multiple shade treatments at different levels of light.
Our three plant species, basil, tomato, and Prunella vulgaris, were subject to sun and shade greenhouse treatments. Both treatments in each species started with 10 seedlings, making the total number of seedlings 60. All 60 individuals were fertilized during week 1 to ensure that all individuals would be of an adequate size by the time measurements were taken in the following weeks. Photosynthetically active radiation was measured with the LiCor Li250 device to determine the difference in light availability between the treatments. The full sun reading was 37.6 lux compared to 0.6 lux in the shade, both measurements taken at 11:30 a.m. Seedlings were allowed to grow under the different treatments for 22 days, after which the growth metrics were measured. Internodal length was measured from the node at the cotyledon leaves to the first node of the plant. We expected the plants in the shade to have a greater distance between the nodes due to them stretching toward any light source. But we found that internodal distance was significantly greater in the sun-grown tomatoes and the sun-grown basil. This is most likely because sun-grown individuals were subject to better conditions that allowed them to grow larger than shade-grown plants in regards to photosynthetic rates and internodal length. There were visual signs of stretching, as the shade-grown plants were lighter in color and their stems did not stand up straight. Prunella vulgaris individuals were of inadequate size to measure internodal length because the sample plants were too young and lacked nodes, which are only present during flowering.
Chlorophyll content was measured using the apogee chlorophyll meter. We expected shade-grown plants to have lower chlorophyll content than the sun-grown plants of the same species. This was the case among all species. Plants grown in direct sunlight had more chloroplasts to take advantage of the high levels of light and increase their photosynthetic rates. Shade plants had lower chloroplast count due to dramatic light limitations. If this experiment was repeated, chlorophyll content would be measured at multiple life stages throughout the life of the plant to understand resource allocation in individuals when faced with lower light levels. Stomatal conductance was measured with the Decagon SC1 pyrometer. We expected stomatal conductance to be higher in the sun-grown plants compared to the shade plants of each species. This expectation was true only for Prunella vulgaris, but we were surprised to find that there was no significant difference between the treatments for tomato or basil. It is likely that within the life histories of these annual agricultural species, they have not experienced low levels of light. Therefore, they continue to transpire at the same rate even when they are in shaded conditions where photosynthetic rates are low, and it would be more beneficial to lower stomatal conductance in order to reduce water loss.
Prunella vulgaris showed signs that it was able to close stomata and reduce water loss in the shade because its stomatal conductance was significantly lower in shaded treatments where photosynthesis rates would be lower. Prunella is a native perennial species, thus it is more likely to have a life history where it would have been exposed to shaded conditions, and therefore more likely to have adaptive plasticity to tolerate shade. Another explanation of these lower stomatal conductance values found in the shaded Prunella individuals could be due to the fact that the plants in the shade were much less healthy and unable to transpire at the same rates.
If If the growth metrics were not significantly different between the shade and sun treatments, then this would suggest that growing these species in the shade would be a possible option for farmers to incorporate tomato and basil plants into agroforestry schemes. Unfortunately, from this experiment, we can conclude that it isn't viable to grow basil and tomato plants using agroforestry methods. Unlike shade-tolerant agricultural plants like coffee, the results from this study show that plant performance of the focal plants significantly decreased. There is no phenotypic plasticity in basil and tomato plants, which is suggested due to the influence of agricultural evolutionary selection. Reiterating what we said before, this study showed that Prunella vulgaris has adaptive plasticity to shade, a possible physiological adaptation to tolerate a wide variety of environmental conditions. Future studies should look into other agricultural crops that can be grown using agroforestry methods, which may be useful in ecological forest management or other applications.